everyone. I'm Dr. Reshma. I'll be talking about cell injury. To begin with cell injury, you should know about the basic function of a cell and structure of cells. Cell is nothing but a basic unit of tissue which form an organ and system in a human body. What happens? Body cells are divided into two main types. First one is epithelial and mesenchymal cells. Virchow was the first person to publish a cellular theory of disease, in which he has told that concept of disease which occurs due to abnormality at the level of cell. Most forms of disease begin with cell injury followed by consequence of cellular function. Coming to definition of cell injury. Cell injury is defined as a variety of stress a cell encounters as a result of changes in its internal and external environment, which means that any cell injury, it occurs due to a stress. It might be due to internal or external stimuli. In general, cells of a body have an inbuilt mechanism to deal with changes in environment to an extent. The cellular response to stress may vary and depend upon the variables. First one is type of cell and the tissue involved in a cell injury, extent and the type of cell injury. Various forms of cellular response to cell injury are as follows. First one, when there is increased functional demand, the cell may adapt to change which may be expressed morphologically and then revert back to normal after stress is removed. Second one, when stress is mild to moderate, the injured cell may recover. It is also known as reversible cell injury. When the injury is persistent, cell death may occur, and this will lead to irreversible cell injury. Residual effects of reversible cell injury may persist in the cell as evidence of cell injury at subcellular level, or metabolites may accumulate within the cell. This is a flow chart which explains about the cell injury and its consequence. First, the normal cells. When there is mild to moderate amount of stress to the cell, it causes a reversible cell injury. That is, it leads to, and these residual effects will be seen within the cells. It leads to degeneration, subcellular alteration and intracellular accumulation. When there is altered demand, that may be increased demand of a cells, it will lead to adaptations. Adaptations may be of various types like atrophy, hypertrophy, hyperplasia, metaplasia, dysplasia. When stress is removed, normal cell will be restored. When severe and persistent stress is there to the cell, it leads to irreversible cell injury and it will in turn leads to cell death. Coming to basics of cell and its cell structures. By electron microscopy has shown that cell membrane or plasma membrane has trilamellar structures which has a total thickness of about 7.5 nm. And biochemically, the cell membrane is composed of phospholipid, glycolipid, cholesterol, protein and carbohydrate. And there are bundles of microfilament along with cytoplasm and proteins of cell membrane may form a projection on the surface of cell, and these are called as microvilli. Here is a photograph of a cell. Here you can see various organelles of the cells, and this outer part is the cell membrane, which is composed of glycolipids, proteins and microvilli tufts. And this is the center part is a nucleus with nucleoli within and a chromatin and rough endoplasmic reticulum in which ribosomes are attached, and a smooth endoplasmic reticulum where ribosomes are not attached. And this is the mitochondria, ribosomes, lysosomes and villi. Now we'll talk about each components of cell. Cell membrane performs the following important functions. First one, selective permeability, that includes diffusion, membrane pump and pinocytosis. And second one, Beer's membrane antigen. Like it helps, these membrane antigen helps in detection of a blood grouping and a transplantation antigen. Third function is possess a cell receptor for cell to cell recognition and communication. Coming to second organelle, nucleus. Nucleus consists of an outer nuclear membrane enclosing a nuclear chromatin and nucleoli. Nuclear membrane consists of two layers of unit membrane which are separated by 40 to 70 nm wide space. Outer layer of nuclear membrane is studded with ribosome and is continuous with the endoplasmic reticulum. And the two layers of nuclear membrane at places are fused to form a circular nuclear pores which are about 50 nanometer in diameter. This nuclear membrane is crossed by several factors which regulates the gene expression and repair the DNA damage as soon as it occurs. This nuclear chromatin is a main substance of nucleus. It is comprised of nuclear chromatin which forms a short piece of thread-like structures called chromosome, which are 23 pairs. Chromosomes are composed of three components. First is DNA, which accounts of 20%, RNA about 10%, and remaining 70% is formed by nuclear protein, which includes histones, neutral protein, acidic protein. DNA of cell is largely contained in the nucleus. Only other place in the cell that contain a small amount of DNA is mitochondria. Next, you should know about nucleolus. Nucleolus may contain one or more rounded bodies called nucleoli. Nucleus is a site of synthesis of ribosomal RNA. Nucleolus is also composed of granules and fibrils presenting a newly synthesized ribosomes. Next, you should know about the cytosol and its organelle. Cytosol is nothing but a cytoplasm. It contains a gel-like ground substance in which organelles of the cells are suspended. These organelles are the site of major enzymatic activities. Major organelles of the cytoplasms are mitochondria, ribosomes, endoplasmic reticulum, Golgi apparatus, and microbodies or peroxisome. Coming to cytoskeleton. Cytoskeleton is composed of microfilament, intermediate filament, microtubules. These are responsible for maintaining cellular forms and movement are collectively referred as cytoskeleton. These microfilaments are nothing but long filamentous structures having 6 to 8 mm in diameter, which are formed of contractile protein, actin, myosin and other materials. Coming to mitochondria. Mitochondria are oval structures and are numerous in metabolic active cells. They are enveloped by two layers of membrane, outer smooth and inner it is folded into incomplete septae or a shelf-like ridges called cristae. Mitochondria performs the important metabolic functions of oxidative phosphorylation. They also have a role in apoptosis. Coming to ribosome. Ribosomes are spherical structures which contain 80 to 85% of cells' RNA, which are present freely in the cytoplasm or it is present in bounded forms which are attached to endoplasmic reticulum. Ribosome synthesis protein by translation of messenger RNA into peptide sequence followed by packing of proteins for a endoplasmic reticulum. Coming to endoplasmic reticulum. It is composed of vesicles and intercommunicating canals. It is composed of unit membrane which is continuous with both nuclear membrane and Golgi apparatus. You should know about the functions of endoplasmic reticulum. Morphologically, there are two types, rough and smooth. In rough endoplasmic, it is called because outer surface is granular due to attachment of ribosomes. And smooth is devoid of ribosomes. Rough and smooth endoplasmic are generally continuous with each other. Smooth endoplasmic reticulum contain many enzymes which metabolize drugs, steroid, cholesterol and carbohydrates. Coming to Golgi apparatus. Golgi apparatus or Golgi complex is generally located close to the nucleus. Morphologically, it appears as vesicles, sacs, or a lamellae, composed of unit membrane and is continuous with endoplasmic reticulum. Golgi apparatus is particularly well developed in exocrine glandular cell. Main functions are synthesis of carbohydrates and complex protein, packing of protein synthesis in rough endoplasmic reticulum into vesicles. Next organism is lysosome. Lysosomes are rounded to oval membrane bound organelles contain powerful lysosomal digestive enzymes. There are three forms of lysosome: primary lysosome, secondary lysosome, residual bodies. In primary lysosomes, or it is also called as storage vacuoles, formed from the various hydrolytic enzymes synthesized by regular rough endoplasmic reticulum and packed in Golgi apparatus. Secondary lysosomes or autophagic vacuoles are formed by fusion of primary lysosomes with a part of damaged or worn out cell component. Residual bodies are ingested materials in the lysosomes, for example, as in lipofuscin. Centrioles or centrosomes. Each cell contain pair of centrioles in the cytoplasm which are close to nucleus and are called centrosomes. Centrioles are the cylindrical structure composed of electron dense, evenly shaped microtubules. They perform the function of formation of cilia and flagella, constitute a mitotic spindle of a fibrillary protein during mitosis. Now, we have dealt about each component of cells and its function. Now we'll go ahead with cell injury and its etiological agents. Etiological agents of cell injury cells may be broadly injured by two major ways. First, by genetic causes and acquired causes. Acquired causes of disease compromises vast majority of common diseases affecting mankind. Based on underlying agent, acquired causes of cell injury can be further categorized. Coming to various genetic causes, it may be due to the developmental defects, error in morphogenesis, cytogenetic defects, chromosomal abnormality, single gene defect, Mendelian defect, multifactorial inherited disorders. These are the various genetic causes of cell injury. Coming to acquired causes of cell injury, first and foremost, very important cause of cell injury is hypoxia and ischemia. Hypoxia and ischemia is nothing but reduction in oxygen supply to the cell due to various causes. Second, physical agent. Third, chemical agent and drugs. Fourth, microbial agents. Fifth, immunological agent. Sixth, nutritional derangement. It may be due to overnutrition or undernutrition. Overnutrition like a complications of obesity, undernutrition like protein energy malnutritions, aging, psychogenic disease, iatrogenic factors, idiopathic disease. In a given situation of cell injury, more than one of the above etiological factors may be involved. These are the two major broad categories of etiological agents of cell injury. First one is genetic and acquired. Coming to discussion of foremost important acquired cause of cell injury, hypoxia and ischemia. Cells of different tissue essentially require oxygen to generate energy and perform a metabolic function. Deficiency of oxygen or hypoxia results in failure to carry out these activities by the cells. In simpler terms, each one of the cell require oxygen. Oxygen is needed to carry out its function. The function may be to generate energy or to perform a metabolic activity of the cells. Once there is deficiency of oxygen or hypoxia, cells fail to do its function like metabolic function and ATP generation. Hypoxia is the most commonest cause of cell injury. And hypoxia results in following mechanism. Most common mechanism of hypoxic cell injury is by reduced supply of blood to cell due to interruption, that is ischemia. However, hypoxia may result from other cause as well, as like disorders of oxygen carrying RBCs, like in anemia, carbon monoxide poisoning, heart disease, lung disease, or increased demand of the tissue. In simpler term, hypoxia may be due to ischemia. Ischemia is due to constriction of blood flow to the cells. Or other indirect causes of hypoxia are anemia, carbon monoxide poisoning, heart disease, lung disease, and increased demand. Next, we should know about physical agents. In physical agent, it may be due to mechanical trauma, as you see in road traffic accident, thermal trauma by heat or cold, electricity or a radiational damage to the cell, as in ultraviolet and ionizing radiation, rapid changes in atmospheric pressure. Third, chemicals and drugs. Chemicals like chemical poisoning like cyanide, arsenic, mercury, strong acid and alkalis, environmental pollutants, insecticides, pesticides, oxygen at high concentration, hypotonic glucose and salt. Social agents such as alcohol, narcotic drugs and therapeutic administration of drugs. Microbial agents, injuries by the microbes which may have like bacteria, viruses, fungi, protozoa, metazoa, rickettsia and other parasites. Coming to immunological agents, immunity is a double-edged sword. It helps in protection and also in a cell injury. It protects the host against the various injurious agent, but it may also turn lethal and causes cell injury, as in a patients with hypersensitivity reaction, anaphylactic reaction, or autoimmune diseases. Coming to nutritional derangements, any deficiency or excess of nutrition may result in nutritional imbalance. Nutritional deficiency may be due to overall deficiency of nutrition, as in starvation or protein energy malnutrition like kwashiorkor, marasmus, or deficiency of the any minerals which leads to anemia or trace elements. Nutritional excess is a problem of affluent society resulting in obesity, atherosclerosis, heart disease, hypertension. Aging, cellular aging or senescence may lead to impaired ability of cells to undergo replication, repair, ultimately lead to cell death. Coming to psychogenetic diseases, there are no specific biochemical or a morphological change in a common acquired mental diseases due to mental stress, strain, anxiety, overwork, frustration, depression, schizophrenia. Problem of drug addiction, alcoholism, smoking results in various organic diseases such as liver damage, chronic bronchitis, lung cancer, peptic ulcers, hypertension, ischemic heart disease. Iatrogenic cause. Although as per Hippocratic oath, every physician is bound not to do or administer anything that causes harm to patient, there are some diseases as well as death attributed to iatrogenic cause. For example, occurrence of disease or death due to error in judgment by the physician and untoward effect of administrative therapy like drugs and radiation. Coming to idiopathic cause. Idiopathic means unknown cause. Finally, although not much of known etiology about the disease. This is about the etiological agents of the cell injury. Now we'll talk about pathogenesis of cell injury. Pathogenesis of cell injury of reversible and irreversible cell injury. Following principles applies in the pathogenesis. Type, duration, severity of the injurious agent. Extent of cell injury depends upon type, duration and severity of the stimulus. For example, Small dose of chemical toxin for a short duration of ischemia causes a reversible cell injury, while a large dose of same chemical agent or persistent ischemia causes a cell death. Coming to type, status and adaptability of the target cells. Type of cells are as regarded its unsusceptibility to injury, its nutritional and metabolic status and adaptation of cell to the hostile environment determines the extent of cell injury. For example, skeletal and cardiac muscles. Skeletal muscles can withdraw hypoxic injury for longer time, whereas cardiac muscles suffer from irreversible injury after 20 to 30 minutes of persistent ischemia. These are the two major principles behind the cell injury. One is type, duration and severity. For example, small dose of chemical toxin for short duration cells can withstand, but persistent for longer duration of same chemical agent or persistent ischemia will lead to cell death. Second is type, status and adaptability of the target cells. In this, the example is skeletal and cardiac muscles. Skeletal muscle can withstand hypoxia for longer duration, whereas cardiac muscles suffers irreversible cell injury after 20 to 30 minutes. Other one is underlying intracellular phenomena. Mitochondrial damage. As we have studied that mitochondria is involved in ATP generation. Once there is cell injury, it leads to mitochondrial damage and there will be depletion of ATP. Cell membrane damage disturbing the metabolic transmembrane exchange. Metabolic transmembrane, it affects the sodium potassium pump and calcium pump and various other pumps which acts through the cell membrane. Release of toxic free radicals. Fourth, morphogenic consequences. All forms of biochemical changes underlying cell injury are expressed in terms of morphological changes. Which have studied ultrastructural changes become apparent, which becomes earlier than the light microscopic alteration. Microscopic changes of reversible cell injury are hydropic swelling, which is nothing but the hydropic change of the cell. Cells become swollen edematous, which appears earlier than the morphological alteration in a cell death. Interruption of blood supply, that is ischemia, and impaired oxygen supply to the tissue are the most common form of cell injury in the human being. Coming to pathogenesis of ischemic and hypoxic injury. Ischemic and hypoxic injury are most common form of cell injury. Underlying intracellular mechanism and ultrastructural changes involved in reversible and irreversible cell injury by hypoxia and ischemia depend upon extent of hypoxia and the type of cells which are involved. Are continuation process. These mechanisms are discussed separately. Coming to reversible cell injury. In reversible cell injury, if the ischemia or a hypoxia of short duration effects may be reversible on rapid restoration of circulation. As we have told, example, in coronary artery occlusion, myocardial contractility, metabolism and ultrastructural are reversed if the circulation is quickly restored. And the sequential biochemical and ultrastructural changes in reversible cell injury are as follows. Decreased in generation of cellular ATP. All living cells require continuous supply of oxygen to generate a ATP, to generate a energy or a ATP, which is essentially required for variety of cellular functions. Cellular functions may be in a form of membrane transport, protein synthesis, lipid synthesis, phospholipid metabolism. ATP in human cells are derived from two major sources. One is by aerobic respiration, second is by anaerobic respiration. In aerobic respiration or oxidative phosphorylation, which require oxygen in mitochondria. In second type, Cells may switch over to anaerobic glycolytic oxidation to maintain a constant supply of ATP in absence of oxygen. Ischemia due to interruption in blood supply as well as hypoxia from other causes limits the supply of oxygen to the cell, thus causing decreased ATP generation from ADP. In ischemia, aerobic respiration as well as glucose availability both are compromised, resulting in more severe and faster effects of cell injury. Ischemic cell injury also causes accumulation of metabolic waste products in the cell. On other hand, hypoxia from other causes like RBC disorder, heart disease, lung disease, anaerobic glycolytic ATP generation continues and thus the cell injury is less severe. Highly specialized cells such as myocardium, proximal tubular cells of kidney, neurons of CNS are dependent solely on aerobic respiration for ATP generation. Thus, these tissues suffer from ill effects or more severely and rapidly. Second, intracellular lactic acidosis. Intracellular lactic acidosis in terms lead to nuclear clumping. Due to low oxygen supply to the cell, aerobic respiration by mitochondria will fail first. This will switch on to anaerobic glycolytic pathway which is required for ATP generation. This results in rapid deletion of glycogen and accumulation of lactic acid. Accumulation of lactic acids lead to lowering of intracellular pH. Early fall in intracellular pH results in clumping of nuclear chromatin. Third, damage of plasma membrane pump. Hydropic swelling and other membrane changes. Lack of ATP interferes in generation of phospholipids from the cellular fatty acids which are required for continuous repair of membrane. This results in damage to the membrane pump operating for regulation of sodium calcium as under. Failure of sodium potassium pump. Normally it requires 3 ATPase. When 3 ATPase are not present, the sodium potassium pump fails at the cell membrane, plasma membrane level, and which leads to accumulation of sodium within the cells. This results in accumulation of sodium and diffusion of potassium out of cells. Accumulation of sodium in the cells leads to increased intracellular water to maintain isoosmotic conditions which lead to hydropic swelling. Failure of calcium pump. Membrane damage causes disturbance in calcium ion exchange across the cell membrane. Excess of calcium moves into the cells, particularly in mitochondria, causing its swelling and deposition of phospholipid rich amorphous densities. Ultrastructural evidence of reversible cell membrane damage is first is loss of microvilli, intramembranous particles, focal projection of cytoplasm which is also known as cytoplasmic bleb formation. Myelin figures may be seen lying in cytoplasm or present outside the cell. Reduced protein synthesis. Dispersed ribosomes as a result of continued hypoxia, membrane of endoplasmic reticulum and Golgi apparatus swells up. Ribosomes get detached from the rough endoplasmic reticulum and polysomes are degraded into monosome, thus dispersing ribosome in the cytoplasm and inactivating their function. In simpler term, due to hydropic change, what happens is membrane of rough endoplasmic reticulum, these ribosomes will get detached and these ribosomes and forms a polysome and polysomes are further degraded into monosome and dispersing. These ribosomes get dispersed in the cytoplasm which inactivates their function. Similar reduced protein synthesis also occur in Golgi apparatus. Withdrawal of acute stress that resulted in reversible cell injury can restore the cell to normal state. This is about the reversible cell injury. Now we'll talk about irreversible cell injury. Persistent ischemia or a hypoxia results in irreversible damage to the structure and function of cell. The stage at this point no return or irreversibility is reached from reversible cell injury is unclear, but the sequence of event is continuous of reversible injured cell. Two essential phenomena always distinguish irreversible form from irreversible cell injury. Inability of cell to reverse mitochondrial dysfunction on reperfusion or restoration of oxygenation. Disturbance in cell membrane function in general and plasma membrane in particular. Further reduction in ATP, continued depletion of protein, reduced intracellular pH, leakage of lysosomal enzymes into the cytoplasm, and these biochemical changes have an effect on ultrastructural components of cell. Coming to first, calcium influx. Mitochondrial damage as a result of continued hypoxia. A large cytosolic influx of calcium ion occurs, especially after reperfusion of irreversible injured cell. Excess of intracellular calcium collects in a mitochondria, disabling its function. Morphologically, mitochondrial changes are appearance of vacuoles in the mitochondria and deposits of amorphous calcium deposits are seen in the mitochondrial matrix. Activation of phospholipase. Membrane damage to membrane function in general and plasma membrane in particular is most important event in a irreversible form of cell injury. In ischemia, a sustained ischemia, there is increased cytosolic influx of calcium in the cells. Increased calcium activates endogenous phospholipase. This in turn degrades the membrane phospholipids progressively, which is the main constituents of lipid bilayer of cell membrane. Besides, there is also decreased replacement synthesis of membrane phospholipid due to reduced ATP generation. Other lytic enzymes which is activated is ATPase, which causes further depletion of ATP. Increased calcium will activate phospholipase, and these phospholipase break down or degrade the membrane phospholipid, which is the major component of lipid cell membrane bilayer, and other enzymes like ATPase is activated, which depletes ATP. Coming to intracellular protease, which leads to cytoskeletal damage. Normal cytoskeleton of cell membrane composed of microfilament tubules and intermediate filaments, which anchors the cell membrane, is damaged due to degradation by activated intracellular protease or by physical effects of cell swelling, producing irreversible cell membrane injury. Activated endonuclease. Nuclear damage. The nuclear proteins are damaged by activated lysosomal enzymes such as protease endonuclease. Irreversible damage to nucleus can be in a three forms: pycnosis, karyorrhexis, karyolysis. Pycnosis is nothing but condensation and clumping of nucleus, which becomes dark basophilic. Karyorrhexis, which is nothing but nuclear fragmentation into bits. Karyolysis is dissolution of nucleus. Damaged DNA activates pro-apoptotic protein, leading to cell death. Third, activation of lysosomal hydrolytic enzyme. Lysosomal membrane are damaged and results in escape of lysosomal hydrolytic enzymes. These enzymes are activated due to lack of oxygen in a cell and acidic pH. These hydrolytic enzymes include hydrolase, RNase, DNase, protease, glycosidase, phosphatase, lipase, amylase, catepsin, which on activation brings about enzymatic digestion of cellular component and hence the cell death. The dead cell is eventually replaced by mass of phospholipids called myelin figures, which are either phagocyted by the macrophages or may leads to formation of calcium soaps. Liberated enzymes just mentioned leak across the abnormal permeable cell membrane into the serum, estimation which leads to clinical parameters of cell death. Like in myocardial infarction, TROP-T, LDH is raised and CKMB. This is the flow chart which explains about the ischemic hypoxic injury. Ischemic hypoxic injury leads to which will hamper ATP generation by two mechanisms: aerobic respiration and anaerobic oxidative phosphorylation, anaerobic glycolytic pathway. When these two are hampered, there is decreased ATP generation. When there is decreased ATP generation, on a cell membrane, ionic pumps are hampered, sodium potassium pump and calcium pump, which leads to increased cytosolic calcium. Increased cytosolic calcium in turn leads to increased mitochondrial calcium, which activates a phospholipase enzyme. This activated phospholipase enzymes degrade the phospholipid which is present on the cell membrane and break down the lipid products, which also activates protease, which causes a cytoskeletal injury and membrane damage. Next is it liberates a toxic oxygen radicals like oxygen superoxide, hydrogen peroxide, hydroxide ion. Liberation of intracellular enzymes will cause nuclear changes like pycnosis, karyolysis, karyorexis, and myelin figures deposition within the mitochondria and cytoplasm. And serum enzymes are liberated like SGOT, LDH, CPKMB, TROP-T. These are the various changes on the organelles of reversible and irreversible injury. In reversible injury, mitochondria undergoes swelling, and on ultrastructural studies you see amorphous dense deposit. Whereas in irreversible cell injury, swollen mitochondria with the vacuoles, and you'll see large amorphous deposits within the mitochondria. Next, on membrane, you'll see in reversible cell injury, you'll see cytoplasmic blebbing, intramembranous deposition of the particles, and cell swelling, myelin figure deposition. Whereas in irreversible cell injury, you'll see disruption of these intramembranous filaments and myelin figure. Coming to rough endoplasmic reticulum and ribosomes. Rough endoplasmic reticulum will swell up and ribosomes will get dispersed, which forms polysome, monosome, and disruption of functions. And in irreversible cell injury, it gets lysed. Lysosomes, it forms a autophagy, whereas in irreversible cell injury, these are swollen and ruptured. Cytoskeleton, it gets aggravated in reversible cell injury, it will be disrupted in irreversible cell injury. Nuclear, you will see clumped chromatin, whereas in irreversible injury, you see pycnosis, karyolysis, karyorexis. This is all about the cell injury. Thank you.